All right, guys, let's get started here. So this is the third lesson on the Diffusion MRI crash course. Today it's going to be all about the quality control. We're going to have general introduction plus some hands-on things, and at the end it's going to be a small demo. Again, as like before, it's about an hour. Okay, so yeah, so it's about Diffusion MRI QC, still a topic, and I mentioned that many times before, that's underappreciated. It's slowly becoming more common, and finally, people really reviewers look in papers whether appropriate QC was done. If you look at papers from 2 or 3 years ago, the norm was no QC was done because people didn't know how to do QC, and most people used FSL. And if you used the old style FSL, there was nothing really in there for QC. Sure, you could look at it, but there were not really good tools to take things out, and there were no automated tools for taking things out. And as soon as you had a little bit more than a few gradient directions, that became not a feasible way of doing things. So today I will talk about these different artifacts, motion eddy occurrences, stability artifacts, and how to correct them. I'll also talk a little bit about the nifty to nerd conversion and DICOM conversion, because that's always a first step. Talk about whether you want to remove DWIs the way we're doing it in our lab, or rather correct diffusion images such as an FSL proposed, as well as at the end of how we do investigative tractography in Slicer and why that's necessary. So I've shown this slide before, I think in the first presentation. This is kind of like an overview of artifacts you may encounter in general in diffusion imaging. Now, there's a lot of them. And of course, in older data sets, there's way more of these. In newer data sets, there's less, but there's not a single case of a data set that I've looked at in all my neuroimaging studies where there's not at least one of these present. So it's common that let's say if you have 100 DWIs you require, then at least 1 up to 10 very commonly will need to be either removed or corrected. So this is an existing and prevalent thing. So it's just there all the time. And we're going to talk about all of these in turn. Like I mentioned before, our view with DTIPrep is to remove bad DWIs. And one can argue that for certain artifacts, one could keep them and interpolate them, whereas for others, you probably don't. The problem is the available tools out there, there's not one that kind of like quantifies the artifacts and says, all right, like this is now too much, let me not interpolate it. It's either all remove, all bad remove, so the way we're doing, or all bad interpolate. And probably something kind of a middle of the road decision where if the artifact is small enough, let's interpolate. If it's too large and too many slices are affected, let's just get rid of the heck. All right. So there's, from a DTIPrep point of view, but also FSL point of view, there's in some sense 2 sets of artifacts that you can see. One is more on a slice by slice approach where you have an individual slice that's just off, maybe a set of slices, but it changes from one slice to the next. Something happens. That can be that it suddenly gets very bright, although that is less observable, but what you observe is suddenly gets very dark, meaning the slice just either goes fully black, so there's no signal, or very, very low signal. That's also called dropout, signal dropout. That's probably the most common artifact that you see in diffusion MRI. DTIPrep goes through the slices one by one, and if the intensities change too much from one slice to the next, it flags it. Basically looks at correlation between the slices. If the correlation is too far off, says, oh, I don't want to use this, this image. There's, I'm mentioning it here because later I actually do not have it in the presentation. There is in DTIPrep something like a head skip and a tail skip, because of course the image can also change just because every slice shows a different anatomy. If the anatomy changes too rapidly, then that falls apart. So you can only do that for really within the brain. As soon as you leave the brain, you may actually get some false positives, which is why DTIPrep has these additional parameters for that. The thing with doing the way DTIPrep does it by throwing out bad TWI data is, let's say we have only one slice that's bad, and all the rest is good. That means for that whole DWI, where we have maybe 50 slices, we throw the whole thing out, even though only one slice is bad. So an alternative view would be, well, maybe we should label that slice as bad, and then we use whatever is good, at least. And just do not use that slice. Or, as it is done in several other processes such as FSL and DTIPrep, actually it detects those slices that are bad and then interpolates those. FSL and DTIPrep both are relatively new development. They've been around now, methodologically wise, for 1 or 2 years. They're actually fairly similar. What is common to them is that the corrupt slice is being interpolated from the other gradient information. So the more gradients you have, the better you can interpolate the data that is bad. But at both steps, you have an identification followed by a replacement, so to say, step. The difference is DTIPrep, which is Guido's tool from NYU. That one uses a spherical harmonic, so a parametric model for interpolating, whereas FSL uses a non-parametric Gaussian process model. I wouldn't necessarily say what one is better than the other. In both cases, though, in order for this to work, that you can use the other gradients, is you have to have applied already all kinds of corrections of motion, eddy current, and susceptibility artifacts. Those have to be corrected, otherwise you would compare apples and oranges. So a voxel in one gradient has to equal the same voxel in the other gradient. It has to actually reflect the same anatomical location. And in order for that to be the case, all these other artifacts, such as motion, eddy current, and susceptibility, have to be solved already. There's another one that I mentioned here, is if you only are interested in doing DTI, rather than solving it up front when you do the QC, you can also solve it on the back end when you're doing the diffusion tensor estimation. So you can do that estimation in a very robust way. That method is called Restore. It's from NIH, from Carlo Pierparoli's group. It's in Turquoise, is the tool called. And it estimates the DTI model with a very nice, robust things that rejects outliers, etc. And it even quantifies those outliers. It provides an outlier map and things like that. That can be if you, so if you don't do any QC, any rejection of gradients, and so you use neither DTIPrep, DTIPrep, or FSL, but only are interested in doing DTI, you could only do Restore and get a decent good things, because it gets rid of all the bads. It assumes that you have more good than bad, of course. Because if you have just a significant amount of bad, that's somewhat, and let's say that badness is correlated, then this falls apart, of course, because it's a non-outlier detection. And yes, and last is, that's how it used to be done. I don't care, you know, head in the sand. Just use the data, even if it's bad, to estimate the tensor or connectivity or anything like that. And this often leads to incorrect results. There's actually a nice paper from Carlo's group where they show how artifacts can lead to incorrect results in studies. So for example, this is applied to pediatric study, I think it's in autism even, where they show that, yes, if you don't do quality control, these are the results you get, which are totally off, because when we actually correctly correct for these artifacts, we get very different results. So, I mean, it's something that's kind of like understood in most other modalities. Like if you see a structural image and it's totally noisy and in motion, you would never use it. But the problem is the diffusion imaging data is not intuitive to us, which is why often we say, oh, I don't really know what to do with it. The tool processes it, I'll take it. I guess it's similar to the early days of functional imaging, which is, again, it's very different nowadays. So I said there's two types of artifacts. One is slice by slice, and the other one is what we call an interlace artifact. The interlace artifact is largely a motion artifact, and that stems from the way the data is acquired. The data is acquired in a 2D EPI acquisition usually. That means the data is such acquired that first the odd and then the even slices are acquired, and that means it starts, let's say, if it goes from top to bottom, it says here 1, 3, 5, all the way to the top, and then once it's done here, it goes back down here and then gets the even ones. Now, if there's motion during the acquisition of this 2D sequence, it looks very different from motion that you would see in a structural in a 3D. If you have a 3D acquisition, for example, for the MPRH, you may be used to looking at, motion usually is seen as Gibbs ringing. So it's ringing around the edges of the brain. Here, motion is usually seen as an initial blind artifact, where kind of like the odd and the even slices just seem off, as if they stem from two different acquisitions that have been incorrectly fused together. So you usually won't be able to see it if you just look at the axial direction, if the axial direction is the acquisition direction, but you would have to look at either the sagittal or the coronal. And there you can see that initial blind artifact very well. Again, FSL and DTIPrep approach these differently. DTIPrep detects those as a special detection for motion, where it basically makes an image of the odds and the even slices. It looks at the correlation between the two. It tries to even register them, and if that registration is successful, it looks like how much translation motion was necessary to register the odds and the even slice image. And if that's too large, it throws it out. It says there's motion within the DWI image. FSL, and again, this is only in the new version of FSL, just like what I mentioned before. Also, the correction of the slice by slice, that's only in the new FSL. And when I say new, like this year new. So here I write 5.12 and 5.13 are the only ones on the 5, and 6.1. If 6.1 is already out, I don't know if the 6.1 is actually out already. So this is super new. The corresponding methods paper came out last year. And what FSL does, just in the whole spirit of rather than rejecting data to try to correct it, it tries to correct this within DWI motion. And in order to do that, it actually has to treat each slice as its independent acquisition. So it will do a slice to volume registration. So it has a volume, how it's supposed to look, and it takes each slice and registers that slice to the overall, how the anatomy should look like. This is nice because you can indeed then correct motion this way. It is bad because 2D to 3D registration is usually ill-suited. It has a lot of errors on the rotational aspects. Particularly depends on the anatomy. At certain locations of the anatomy, you have better accuracy of the registration than in other locations of the anatomy. And so I generally would say you should stay away from this until we have good results that shows that this is consistent and indeed valid. I would not use it. And also, it's super, super slow. I mean, in generally, with this new version of FSL, it's going to be slow, but this probably doubles the computation time, if not more. Rather, if you have indeed bad motion in your data, remove those. There are not going to be many of those usually there. This is not the main artifact you encounter. You'll have a few of these. The main artifact you encounter is the slice-wise artifact, not the within DWI motion artifact, so the interleaved artifact. All right, so we have a whole slew of slice-wise artifacts. We have motion artifacts. We can either correct them or we can throw them out. So now we can use either DTIPrep or FSL to do all these kinds of things. Now, if I'll put it together, what would a modern or up-to-date diffusion MRI QC workflow would look like? Now, this means modern meaning today. If I would be given this talk last October, this would have looked different. So this is updated because we have the new version of FSL. Well, first is DICOM conversion. That's always the case, and I'll have a slide on that. And then usually it would be best to first go and throw out the bad stuff. So the best practice would actually be to run DTIPrep, get the stuff that's really bad, and throw it out. So you could run DTIPrep more with a conservative threshold on all these things, so that you say, well, maybe I'm going to interpolate later something with FSL, but only the stuff that's not truly bad. The reason why I say better here, you see down the road, it comes down here again, it's as simpler, is because a lot of these settings here become harder to do if you remove stuff here, because now for every subject you have a different number of gradients, and so you have to adjust all your configuration file on a per subject level. Definitely possible. We don't have the script for that yet done, which is why we don't do it. But if you want to do the script, be my guest. This is the better thing to do, but if you do it at the end, it's probably fine too. I don't think you actually lose much by doing it later. You're not introducing a lot of stuff by doing it later. But anyway, so once you have the bad TWS removed, or you choose to do it later, the first thing to do is to correct the geometry. And that is the susceptibility artifact computation. We'll go into that right away after that, where we remove the geometric distortion that is inherent in all of our diffusion images. Then we create a brain mask, very much necessary for the next step, for the eddy current and motion correction. The reason why the brain mask is necessary is if we want to include the susceptibility artifact correction the right way, we have to do it jointly with the motion estimation. So we can't just apply this susceptibility artifact computation, because that's a distortion, geometric distortion, that's relevant to the anatomy. So when the subject moves, it moves with the anatomy. So that's why you have to correct motion and apply this undistortion field at the same time. That's, by the way, only possible with the new FSL. The old FSL, you had to assume that the motion was small enough that if you're doing this, it's okay. It's okay to do it, assuming that there's not enough motion. So after the motion correction, you would then do any kind of stuff down the road. You can estimate your tensors. We always, as part of our QC, estimate tensors, even if that's not the goal for the analysis, because from the tensors, we do investigative tractography to see whether we can determine any other problems with the diffusion scan that we were not able to pick up before, such as vibrational artifacts or any kind of issues with scanner electronics. If you have scanner electronics, let's say you have an issue with one of the gradient or the electronics of one of the gradients, you won't be able to see it anything before. But when you then go to the tractography, you will see it immediately. All right, so that's kind of like the makeup, and I'm going to talk about each of these steps. First, DICOM conversion. The first thing is that this is still a big mess. So DICOM is supposed to solve standardization across vendors. And it has done a great job in many imaging modalities. Diffusion imaging is still not that great. There are no standardized tags for the prescribed protocol, meaning what protocol was planned. So what's the p-value supposed to be, and what is the gradient direction supposed to be? That's more or less standardized nowadays. Still, depending on the vendor may not provide a DICOM that's standardized, so then you have a problem there. But yeah, so you need a good conversion tool that can handle multiple vendors because of that. There's not a single great tool, but there's two good tools that I can recommend. One is DWI Convert. It's in Slicer. So I usually run it on the command line, but you can run it also on the Slicer GUI if you prefer that. I think it's the best one at the moment because it supports the most vendors, and it is an appropriate reflection of the measurement frame if the measurement frame is off from the patient coordinate system in the scanner. This can happen particularly with some Philips scanners. So with some Philips scanners, if you use DICOM to NIX, actually it should be 2i, DICOM to NIX, you can get into problems because the gradients are in DICOM header are not specified in the image space, but rather in the scanner space. And so there has to be a conversion of those gradient directions into the image space. And that is difficult to do if you're not capturing, and DWI Convert captures as well. Now here at UNC, you don't need to care. We don't have Philips scanners. We have Siemens scanners. Another thing that DWI Convert does nicely is it has an option that you can tell it to not use the protocol gradient information, but the actually applied gradient information. So the gradient that we're actually measuring diffusion along is not exactly the gradient that was planned. You can't do it exactly along that direction. It's usually just a tiny bit off. And the b-value is also a tiny bit off. So for example, if you say I want to get a b-value 1000, what you're actually getting is probably a b-value of 998 or 1003. Often that really doesn't make a big difference in any of the computations. But nicely here, this tool gives you the option to go to the accurate reflection of what was applied. So what comes out of here is then with this B-matrix approach is that you actually have for every gradient the right direction and the right b-value. DICOM to NIX does not give you that. Even if you only use FSL, I suggest you use DWI Convert because it can also go directly from DICOM to NIFTY. You don't have to go to NERD, even though NERD is its preferred format. It also allows you to go back and forth between NERD and NIFTY. And I have quickly 2 slides on NERD versus NIFTY. So those are the 2 dominant formats in the field. Although I have to say, when I say 2, NIFTY is by far the more dominant in the field because of FSL and its dominance. NIFTY is an okay file format. The problem that I have with NIFTY is that it's a distributed file format, that its information on the gradients are not in the image header. And so theoretically, particularly if you store multiple files with different b-values and b-vectors in the same folder, it's very easy for it to become confused. And another thing that makes it hard is even the default on FSL is that these b-values and b-vec files don't have to have the same name as your images. Whereas, for example, in other formats where the header may be distributed, that often has to be the case. So that in some sense, yes, the suffix may change where you have the different information, but at least the name of the files otherwise has to be the same. That's not the case here. So it's a little bit more room for error that can come in. Another thing, by way how the 2 are different, is that the b-vectors are always stored in image coordinate space. So you have to be sure that that's correct. Whereas, like I mentioned before with Philips, that some vendors have the gradient stored in a different space. So either during the DICOM conversion, you can basically do this transform of the gradients from the usually the world coordinate system to the image coordinate system, and then you can do it during the import. But if it's not possible during the import, you can also do it in the NERD header by specifying a measurement frame. So basically you're specifying an additional transform, and you can then leave your gradients the way they come from the DICOM header. Measurement frame, so this is basically just the information how it's reflected in the NERD header, where you have the standard transform that also Nifty has, that takes the image space to the world coordinate space or patient coordinate space. And in the NERD, you have an additional measurement framework that takes the local information that you have as it's measured to this world or patient coordinate system. Another thing that I like about the NERDs is that all this stuff is all in one header. Everything is together. I like that. And that header is ASCII readable. Now that may sound like something really small, but I really like being able to just use an editor, look at the header, and see all the information that needs to be there. Sure, in Nifty I can have FSL as a tool, FSL HD, and it's supposed to give me the full header, but I have to trust it that it gives me the full header. Whereas on NERD, I know what the full header is because I'm directly looking at it. But they're both, from a viewpoint of what they can capture, what they can do, they're both equally powerful. All right. And yeah, and again, if you want to go back and forth, DWI Convert is the tool. Anything in Slicer uses only NERD. Anything in FSL uses only Nifty. So if you ever use Slicer and FSL, you'll definitely need to go back and forth between the two things. All right, so much about the conversion and the file formats. Now let's talk about the first two steps of the corrections. So this is assuming now we've gotten rid of the bad stuff already. Now the first thing is to correct for any geometric distortions that we have in the data that's consistent for all the DWI. And that's the susceptibility artifacts. Susceptibility artifacts, they usually arise from in the brain through air tissue interfaces, such as closeness to the sinuses. So that's why you see in the prefrontal lobe, you see usually big differences. So this is an AP polarity image, and this is a PA. You can see how that is different. Where this kind of like, look, is the ground truth. That's the real anatomy. It is stretched out or scrunched together. Now, if you look on this end here, you don't see really much of a difference because there's no air tissue really close by. Now, if you would have a metal plate in your head, you would see similar things. So it's not just air tissue. It's all kinds of interfaces or non-tissue, basically non-water containing elements versus water containing elements. That interface will always have a problem. And like I mentioned before, this is relevant to the anatomy. So it's not from the coil or the scanner. Of course, it relates to it as well, but it is from the anatomy. So if the subject moves, this moves with the subject. And yes, at first approximation, this is constant for all the DWI. And it's important to say at first approximation. It actually does change a little bit from based on which diffusion direction and on which, but it's only second order functions. And nobody, at the moment at least, goes beyond that to say, okay, it's the same, and I'm applying the same correction for every DWI. Will probably also be just too complex if you want to incorporate a DWI-specific susceptibility artifact correction. There is an alternative to this having to correct is up front on the scanner, you can have a sequence that does a twice refocusing. So that does AP in the first, it applies another echo, and there's the PA in the other, and the two things cancel each other out. And then you're getting basically a near geometrically corrected image out, and you don't need to worry about this. Some of our early studies always use this. That's great. You don't have to worry then. The problem is that you need to scan for longer because the SNR is lower, which is why most modern sequences do this APPA, because they say, yeah, I can solve it afterwards through top-up or other susceptibility artifact corrections. And so to correct this, if we want to do this post-scan, we need two polarity images. So usually it's APPA or left, right, right, left. And APPA has become the dominant way to encode this just because I think it has mainly to do with the correction tools. Our correction tools work better if the deformation is front-back than if it's left-right. That distorts the midline. It just is, from a tool perspective, seemingly better correction. So, an alternative, if you do not have the other polarity, is you can use a structural image. In this case, the T2 image. That's imperfect because you didn't use exactly the same sequence. You have different echo times and so on, but it's better than no geometry correction. Remember, what we're trying to do is just make the geometry look better, more like the structural geometry. It's not really going to change any of our FA values if we do tensor or anything like that, because it's correcting geometry for all of them. And FSL actually allows you to do both of these, whether you have APPA, so opposing polarity, or whether you want to put in there a structural image. What of course you need to do is you need to take that structural image and register it rigidly to your data first, and then once you have that, then you can use it as an input into top-up. This is relatively time-consuming, so keep that in mind. The good thing is it doesn't have to be computed on all the data. It's computed only on the B0 fields, on the B0 images. So that is why it's not super expensive compared to all the other stuff that will come down the road. When you apply it, we need to apply it with a motion correction. That's the thing that you have to keep in mind. So I always hesitate to use FSL top-up for our old way to approach for old pipelines, because their FSL, the old FSL was not able to do this. And we know we have always motion in our data. So then I just basically do not correct for it. And I'd rather accept that my DWI image is imperfect with respect to its geometry. But that's better than correcting the geometry, but doing incorrectly so. Also, if most of our studies, we have to do a quick QC where people need to know within a very short period whether the scan is good or not. We don't apply sensibility artifact correction for that step just because it's not necessary. It's just it looks better and maybe processing down the road is better, but it's not necessary for the quality of the data. When you use top-up, it's a rather simple call. You give it the B0s, out comes the field coefficients and the field, and those information will then be input into the eddy current and motion correction. And you have two parameter files that you need to provide. One is this FSL rec parameter file. You can just use whatever. If FSL has one that comes distributed with it, you can just use that. There's no reason really for you to adjust it. These are default values of how to compute this undistortion field. It's a deform registration process, and this is the default values work decently well. I'm actually never looked into it for our monkeys whether we could optimize these, because obviously these have been optimized for the human setting.
So even for the T2, I didn't look at that. So do you have to change anything for the T2?
I don't think so. I mean, because it's more related to resolution and so on. Yeah. And when you use the T2 as a target for the undistortion, what you need to do is you need to register the T2 and resample the T2 in the space of the DWI. So now it has the resolution of the DWI. So in that sense, it shouldn't matter. Then it has an additional parameter setting, and that is for it to know which are the opposing polarity images. Which image is one polarity versus the other polarity, and it has to know which axis to look along, whether it's X, Y, or Z axis. So it has this basically has one line per B0 that you have in your image. So this would be, you have 4 B0s in this example, 2 AP and 2 PA. You put them all into one volume. You have now one volume with all these 4 images. And it's AP, so that means that's the second axis, because data is in RAS. So first right, left, then AP, and then up, down. So if you have an RLLR, you would have the 1 or -1 in the first, and the other 2 would be 0. The actual sign is irrelevant, but the relative sign is important. So I could write here -1, -1, 1, 1, and I would get exactly the same result. But obviously it's important that these 2 signs are the same, and these 2 signs are opposite of these 2 signs, because that's what the image polarity encapsulates. There's a last value here, that's the readout time. There's some very nice tutorials you can find online how to extract that readout time from your scanner stuff. So you can find the information in the DICOM header, from all kinds of information there. You can compute that readout time, but you don't need to, because that readout time, unless you have a really special protocol, will be the same for all your data. And since it's the same for all your data, you could set this to 1, and it's all fine. Here it says it to 0.1, but it doesn't really matter. I set it to 0.1, so not to confuse with the other 3, which only are 0, 1, -1.
I just ask a question, make sure I understand what you just said about the AP and PA ordering. What I heard you say is that it's irrelevant whether the PA or the AP come first.
No, that's it's not just is it unimportant whether the AP or the PA comes first. It doesn't matter whether you make the APA 1 or whether you make the APA -1. It just means what you have to encode is that they are opposing each other. And you have to correctly identify the axis where the polarity is done. So yeah. All right. So then TopUp computes this undeformation field. So it's not really a deformation field, it's to undeform it. There is an alternative where you can compute a field map. And from that field map, it's basically compute the undeformation field. FSL has means to do that. I'm not going to talk about that here because that's actually something that we don't use often. If you have data like this, it's really better to actually run TopUp. The nice thing about when you do this TopUp computation is it incorporates a motion correction of the B0s. In order to do that correctly, it has to do that. But now you have a motion corrected B0 image, a TopUp that goes along with that. But if you would like to apply it correctly for motion corrected to the other data, you need to correct the other data also for motion. And that's where then the next step comes in. That's where we need to do the eddy current, motion correction, and application of TopUp correction at the same time. So the old style, this apply TopUp, that's still distributed, that would be not doing motion correction. And yeah, you better have sedated scans or something like that if you want to do this. Otherwise, I think just don't do it. As I said, because often we do not necessarily need to correct for geometry. So yeah, so there's a new style, eddy open MP, jointly estimates motion, eddy current correction as well as visibility artifact correction. It's very slow, so use Longleaf if you can. Yes?
I was reading that there's a newer version of eddy. It's just called eddy And then there's Eddie CUDA, which can run on graphics cards. Which
So
speeds up considerably.
So Eddie OpenNP and Eddie CUDA are the same thing.
Mm-hmm.
Just Eddie CUDA runs on GPUs, whereas Eddie OpenNP runs on CPUs. If you are running stuff on Longleaf, what you realize right now that everybody wants to do machine learning, which means the GPUs are the thing that is the hardest to come by. So yes, if you find that the GPU nodes are not the GPU nodes are not taken, definitely use Eddie CUDA. If the GPU nodes are taken, use Eddie OpenNP. I do not know enough about the implementation to know whether you should actually fuse one versus the other. What we know from our own development, it's very hard to develop two things to be exactly the same. And it's very likely that the two implementations are just a tiny fraction off, which can introduce biases, which you can later get out in the analysis. So if you use Eddie CUDA, do everything with Eddie CUDA. If you use Eddie OpenNP, do everything with OpenNP. It's safer not to fuse them. Also, if you do Eddie OpenNP on Longleaf, do everything on Longleaf with the same version, not one from 6.1 and one from 5.13. Even if they say they're the same, it's best not to, because they may also use other libraries that may be different. And if you go, let's say you have also your local servers, don't mix stuff from Longleaf with your local servers because the systems are likely different. And a compilation on one system versus on the other system will yield slightly different results. Same thing applies, let's say, if you would process something on the Mac as well as something on the Linux box. Don't fuse those results. That's just general things. There's always biases. Sorry. All right. So yeah, for running Eddie OpenNP, Eddie CUDA, for doing all this jointly thing together, to do this the right way, you need a brain mask. The brain mask really, that necessity only came in with the new version of FSL. Currently, our version of DTIPrep does not need a brain mask, but that's probably also the reason why FSL's version of motion correction works better. And it works better on the high B value images. It works about equally well, or I think in part even better on our end, for the low B value images. And the reason is that as soon as you go to the high B value images, the background becomes so strong that it can influence your registration. And you don't want that to happen. That's why the brain mask is there. Now, this brain mask does not need to be perfect. It just basically needs to tell the image where to focus on when it does the correction versus where not to focus on. But if you're doing the brain masking already, and you may need it later on for other stuff, sure, you can do it the right way and even manually correct it, so it's done at once, and then you're fine. But for the purpose of QC, you can also have an automatically computed brain mask and just use that. Often that is perfectly fine. You don't need more than that. There's really two ways to do that. If you have structural MRI that you have already brain masked, you can propagate that structural MRI to the geometry corrected average B0. Remember, when you run top up, you actually get out a B0, an average B0 image. It's not average, just but the set of B0 image that have been corrected for top up, for the susceptibility artifact. So now you can take this structural MRI image and do a registration to the diffusion image. It does not correct for all geometry distortions. So if you just do a rigid registration, you're not going to be perfectly aligning things. We usually just do an affine registration for that part. And again, this is, like I mentioned, this doesn't have to be perfect. It just has to basically say where is about the brain and where is the rest. If you look at FSL best practices on their FSL wiki, it actually shows you how to create an average B0 from the top-up output, and it has a thing where it applies SPET to that average B0 and then gives an image out. And so that is pretty decent. I suggest if you do that, is not to use the average B0, but we actually use the average IDWI, isotropic DWI, which is the geometric mean of all the diffusion images. The nice thing about the IDWI is, this is an IDWI image. You can see that there's hardly anything there but the brain tissue. It's so good that nearly you can do a threshold and you get a nice brain mask. It's not, you need to do more than a threshold, particularly further down here with eyes and other tissues, you can have some issues with that. But if you apply FSL bet to the IDWI, you get a very nice result. So you can do what FSL things do it, which is apply to the B0. I suggest IDWI, which is very easy to compute in FSL or in DTIPrep. And that gives you now this brain mask, which is the input for the next step. And I mentioned that before, if you want to correct this for all kinds of other processing down the road, this is easy to improve. So just load it in ITK Snap or in your favorite editor and improve it. What I always say, look at it with the FA as well, so that you can see whether the skull has been fully removed. Because the skull, since it has no signal on the FA, looks really bright. The FA is very noisy in areas of the background and the skull as well. So this way you can clearly see whether the skull is fully out. Whereas if you look at the B0 or the IDWI, the skull is just dark. It's actually easier to see or remove stuff that's bright than stuff that's dark.
A quick question about this first option. What tool would propagating SMR?
You can use, if you're using FSL, you can use FLIRT. In Slicer, you would use the general registration tool in Slicer, which uses brains fit underneath. So that gives us the brain mask that we can now put into the estimation of the motion and eddy current correction. So one thing about the motion correction is not just do you want to correct for it, but you want to report it. So both FSL and DTIPrep report it, although FSL things are a little bit hidden away, you have to go search for it. But many reviewers nowadays will ask you, if you haven't done so, to do an analysis of your data versus motion, meaning whether the presence of motion impacts your study. And if so, they want you to correct for it. So they want some way for you to quantify motion and incorporate that in the analysis, or show pre-analysis that your groups are equally affected by motion. The nice thing in DTIPrep is you get a really nice summary out where you can go per DWI, you get a total rotation and total translation out that you can use for deriving anything that you want to do here. For example, in our studies, we take the total rotation and say anything higher than 1% says high motion or motion present. Anything higher than 2 degrees is high motion, and the same thing on the millimeters. And this way you can have kind of like a classification whether an individual DWI has a large amount of motion, middle amount of motion, or no motion. And then you have an additional step, because you have to, this is now per DWI, now you have to summarize it for the whole diffusion thing. So then we do usually the number of DWIs that show such a thing as an enlarged motion. And that number is what we correlate with groups to see whether our groups, one group showed more motion or not. You can do something similar in FSL. In FSL, you get the matrices out. So it takes a little bit more math to go from that to what I just mentioned that the DTIPrep is. It's not difficult, but it takes a little bit of math. Also, with motion correction, again, I'm pointing to the new FSL. Best do it in the new FSL. The old FSL, what it would do is would motion correct everything to the first B0, which means that DWI volumes that are later in the acquisition would actually get further and further away, because motion is correlated, from the first B0. So your first images would actually be easier to correct for motion than your later images. There was actually a very nice article in 2015 by Craigham et al. So this is not from our group, where they compared DTIPrep with the old FSL, and they showed that if you do it the way it is in DTIPrep, you get much more signal out, and you get more sensitivity if you do studies. The way DTIPrep does it, rather than to the first B0, it computes from all B0s an average, an unbiased average. And usually if the motion is correlated, that's kind of like means it's a B0 more in the middle, and then registers everything to that B0. Now the new FSL does it even smarter. I'll show that.
Is
Yeah.
all of this done in Eddy, or is this an additional step after Eddy?
This is old Eddy.
Okay.
This is old Eddy, and in DTIPrep, it kind of has its own Eddy version, where it does it, but it does it through this average B0 that it first computes. And obviously in the new, it's also in the Eddy OpenMP. And Eddy current artifacts are usually solved with that motion correction. So motion correction would be a rigid registration. The thing is, there's additional Eddy current artifacts which induce a shearing of the image, which means that is not something you can correct with motion correction, which is rigid, and also you wouldn't need really a deformation. You would have a low-level deformation that you can basically solve with an affine registration. And as soon as you do an affine registration to correct for eddy currents, you've already done your motion correction as well. So that's the nice thing. So just doing affine registration solves both motion and eddy currents. So virtually all tools out there that do motion correction, whether that's FSL or DTIPrep, do it this way. And also that this assumes that there's nearly no eddy current artifacts on the B0s, because that's where you register to. And again, this is first order approximation. That's true on the first order approximation. So in the new FSL Eddy, how this is all handled, and kind of like this, the reason why I have a separate slide is because everything goes in one. So we have now an estimation of the susceptibility artifact on the formation field, and we have a brain mask, and now we can put this all in this new Eddy, where it does motion correction, eddy current correction. It applies correctly this undistortion from the susceptibility artifact because it follows how the anatomy gets corrected for motion, as well as you can enable the within DWI motion correction. So this slice to volume correction, if you want. I say do not use. Or and in as well, you can also do this dropout correction where it detects bad slices and interpolates those. I prefer removal, but I understand if you enable this, particularly if you've removed the super bad DWIs to begin with. And all of this is done together, which is why this really is super slow. So if you run this in a single core, this is going to run longer than a day. And this is running longer than a day without the these two steps. If you enable these two steps, it's probably going to be running several days, which is why it's so important either to have the CUDA, where you go on the GPU, or use multiple GPUs, multiple CPUs. So what I usually do is I run this on long wave. There is this environment variable called OMP number of threads that tells FSL how many threads to use. So in this case, I use 4. So this is an environment variable that you set before you started. And that speeds it up by a factor of 4. Of course, when you submit stuff to Longleaf, you have to tell Longleaf you're going to be using 4 cores. Otherwise, your job is going to get killed by the scheduler. Another thing that you now need to provide that was not needed to be provided before with the old Eddy is you need to provide the p-value information, the VVEX information, the tuple parameters need to be provided, and the information that the data is shelled. If you have single shell data, you don't need to do that. If you have multi-shell data, if you get this, usually you get crap out of it. So the description says it tries to infer this and find it, but it's not very good at that. So force it to, if you have multi-shell data, that you enable this.
I'm sorry, shell.
Shell means that you would have not just one b-value.
Oh, okay.
So, sorry. So yeah, so most studies nowadays have 2 or 3 shells where you have a b-value maybe around 1000, and then either a low as well as a high one as well. If you only have one b-value in your whole sequence, you don't need to worry about this. In addition, it needs a new configuration file, and that's this motion correction. Rather than correcting just to the first B0, it has done something much smarter. You can do the old style just to the first B0 by just having in this new configuration file all once. What you put in there is the index, base 1, of the volume that you want to register to for the motion correction. So, for example, if I have 5 B0s on a 35 directional scan, and they're all equally spaced, what I could do is the 1 to the 1, of course, that's registering to itself. Then I do all the 1s here. That means all of these go to this one. Then I do all the 2s, then all the 3s, 4s, and 5s.
Maybe,
Yeah.
I mentioned last week or the week before, in our sequences, the ones for Sarah's study, for instance,
Yeah.
the first 2 volumes are B0
Mm-hmm.
images.
It's quite common. So what
in
you
that
would
case,
then
it'd be
do,
1, then 1 followed by 7 or 8 2s.
exactly.
And then
So,
if you have a P concatenated to the PA concatenated to the AP or vice versa, in the middle of that, you'll have just one number followed by
mhm.
a pattern that looks like that.
So putting this together can be non-trivial. You have to really, because like he mentioned, when we then usually run any OpenMP, we're having our PA, we're having our AP, we concatenate them together. So we have one big image. So now we have to all specify this in this one big image. It's not difficult, but you have to do it with smartness. If you want to say, okay, I don't really care, because my subjects are sedated, then just put 1 everywhere. If your subjects are not sedated, put a little bit of time in this, and then you don't have to worry about it afterwards. In all of this, as you can see now in the mention, like both this top-up parameter file as well as this index file, if you do DTIPrep first, or if you throw out gradients first, you'll have to change this for every of your scans. That's why I'm saying one is better, the other one is simpler, because that makes it non-simple. Now you actually have to have a program that generates this, or you have to do manually each time, and if you do manually each time, you may introduce quickly an error, so I do not suggest you do this manually each time. If it's not something that comes with a tool, and currently there is no tool for it, I suggest you do not upfront removal of DWIs, but later on. Yes?
When you concatenate the AP and PA, you also concatenate the B-val and B-vax together.
Yes.
Right? And so from that, could you, like from the B-val, you could write a little program to get the registration targets from there?
Yes, you could.
Okay.
You could. I'm not saying it's actually hard to do, but somebody has to invest the time and the thinking that it works. And particularly, so if you want to do this one here, within DW dropout correction, I suggest you do invest that time to write such a script, because you really, some of the TWIs are like 90% bad slices, and 10% seemingly good slices. And probably those 10% are bad too, but those 10% are going to be used for anything, any computation. It's much safer to just recheck the whole thing. So yeah, if you want to enable this, make sure you get rid of the super bad ones first, and then you need to adapt this stuff here. Or you just don't enable this and then run DTIPrep as the next step that gets rid of all the bad stuff. That's what we're doing right now. So we don't have to worry about this kind of thing, and we can keep this for the same for all of our data. I'm having three more slices, and then we're going to be finished for today. We won't be covering the investigative tractography because there's not enough time, but we'll get back to this. We'll have like 10 minutes of the next session where we're going to be coming back to this. In DTIPrep, what is nice, it has a GUI and a command line interface. In the GUI, you can put together your protocol of what you want to apply, how you want to set your parameters and everything like that. You can do so in interactive fashion, save it. You can run it from the GUI, or you can exit the GUI and then run it on the command line if you want that. It does a lot of things. The three major things that are important nowadays are these slice-wise artifact detection, the motion artifact detection, as well as an additional vibrational directional artifact detection that I haven't yet talked about. I think eddy motion and eddy current correction is done now better in FSL nowadays, particularly with high B values. So that's why I haven't highlighted that one. In our own processing, our updated processing, we're only doing these three. Or if we don't worry about this, we're only doing these two. Yeah, one thing I wanted to show once more is it has also a nice visualization of your protocol of the gradient directions. Now, particularly if it's a single shell, I mean 1B value, this is very nice to look at. You have multiple shells, it's a bit harder to look at, but still you can see whether your data is uniformly distributed. So particularly if you're starting a new study, a new sequence, don't just assume this is good, but load your first scan that you get into DTIPrep, look at this 3D view and make sure that it looks more or less uniform, because otherwise you can have some really bad errors later on. All right, so that's it for today. We can do some more questions if there are more questions. Otherwise, get the questions for next week, or not next, for the next session, where we can come back to, if this now runs all through, how do we do the final step of the quality control? All right.